Reaching the end of the symposium, and now Mario Serrano will present this title. So thank you, Mario.
For the delay, we have a problem with the blockage of the transportation, but we can start it. And first of all, I would like to thank you, all of you, to be so nice, to make this meeting very nice, and to participate. So let's hope we can do it again, and we will do it again. So today I will talk about this project. This is the characterization of the micro and macro transcriptomic responses against this pathogen, Botrytis cinerea, induced by the plant cuticle, or change in the plant cuticle. So the end goal of all of us who are working with the plants is really to produce more and better food. Yeah, and this task is not new. Actually, has been the evolution has been driven by the number of the people who live in the world. So every time that this increase in the population, there is a technological change. There have to be a technological change that here I summarize it. So in this revolution, let's say, starting with the Neolithic, when the agriculture started to exist, then it was okay because there were not so much people, but this was increasing and increasing until there was a necessity to have an agricultural revolution where they start to use the animals to prepare the soil, among many other things. Then in the 1960s, there was a machine revolution, or the green revolution, where they start to use not only the machines, but also fertilizers, new varieties of the seeds, and so on. And then in 2000s, when the DNA was really going to the sequencing of the different plant species, so there was the biotechnological revolution. And the last one is where we are now, and this is the high-tech agriculture using drones and many other things. So with all of these improvements, or technological improvements, we can say that right now we have enough food, even though there is not everybody has enough food to eat, because there's a very big and multifactorial problem. But from the production side, we have enough. Yeah. But then what is the problem? One of the problems is that only during post-harvest, 25 to 50% of the production is lost. So this is really quite a lot of amount of loss of already to eat food. And why? Once more, there is a multifactorial problem because it's the transportation, how to keep the food, but one of the problems are the microorganisms. Among them, there are these species, Alternaria, Aspergillus, Fusarium, Geotrichum, Glosporium, Penicillium mucor, and Botrytis cinerea. So my talk will be focused on Botrytis cinerea. This is the pathogen that we work with. So Botrytis, well, Mario already saved me a lot of my introduction, but I will repeat it because I have it. So Botrytis cinerea is a necrotrophic fungus that affects many different crops, many of them. In the fruits, in the leaves, and also on the flowers and on the leaves. So this is a really big problem. This is the second most important pathogen in the agriculture, Botrytis, and that's why it's important also to study and to try to find out some ways to stop or to reduce the damage that they cause. So how is the process of Botrytis cinerea infection? Well, here is very summarized and very simplified model. So the spores of Botrytis arrive on the surface of the fruits or the leaves, and they deal with the constitutive defense, that is the cuticle and the cell wall. Botrytis, there's not a problem for Botrytis because they have many enzymes and many physical ways to break down these barriers. And once they break down, there is the release of chemicals, the MAMs and DAMs. So once these molecules are released, then the communication starts with the plant because they are recognized by the plant. These molecules are recognized by the plant, but also by the microbe. Once they are recognized by the plant, there is the induction, rapid induction within the seconds of the innate immune response, including the reactive oxygen species and the accumulation of small RNAs. Afterwards, let's say in hours to days, there is the induction of the late defense responses, including the accumulation or the triggering of the signaling cascades dependent of salicylic acid, jasmonic acid, ethylene, and abscisic acid. So one we expect that with all of these defense response, botrytis cinerea infection should be stopped, but it's not. For most of the plants, it's not. They are susceptible. Why? Because botrytis, as yesterday Mario also said, the botrytis is very tricky pathogen and very intelligent, let's say, because they use effectors to modify or to inhibit the innate immune response, such as the oxalic acid, and also because botrytis use the small RNAs also to inhibit the different responses. Okay, what would you like to know or to study in my group, or to try to answer, or to try to identify or to try to identify in this process? Well, first of all, we would like to know what happened at the cuticular level. Yeah? How are released the DAMs, the MAMs, which ones are, how they are recognized. Also, we are pursuing that part. Additionally, we wanted to know how is regulate the innate immune response, including the reactive oxygen species. What is the role of the small RNAs, and how we can get resistance in the end. Yeah. So to try to answer that, we have several projects in my group, including the study of cuticular mutants. Also, what happened when in these cuticular mutants, what happened with the philosphere and endosphere? Yeah, what are the changes? Because we know that these microbes also can modify the different responses and also the interaction with different other microbes, such as botrytis cinerea. Additionally, we are performing a chemical genomics to modify the reactive oxygen species. We are analyzing the small RNAs. We also have some mutants with, that modify the early response genes that are just before the activation of the innate immune response, and mutants with the late defense responses, such as the ones involved in the salicylic acid, jasmonic acid related signaling pathways. And also we have another project that can be modified any of these parts, that is the identification of characterization of biostimulants. So in parentheses, you can find the number of the posters that we have presented each one of these projects. So, but today I will not talk about all of them. It will be impossible. I will just focus on the analysis of the RNA and the small RNAs and the cuticle. So, the plant cuticle and botrytis. So we know that the cuticle is composed mostly by the waxes that could be epicuticular and intracuticular waxes, and the cutin. More than 90% of the cuticle is formed by these two polymers. Then also we have described something that it was already well known, that the cuticle, and you can expect, not only is a barrier that can be involved in the diffusion of gases or liquids, but also regulates, as one might expect, the interaction with herbivores, bacteria, and fungi. So, in the next slides, we have been working with this since I was in Switzerland, that change in the cutin composition leads to the resistance against botrytis. This is a strange idea because we found that, well, I have just said that the cuticle is a barrier. So when you modify, you might expect that the plants are susceptible. Well, it happens the contrary. For some of the plants, the changes in the cutin and the cuticle leads to the resistance. I will explain why we have this. So the first evidence is that when we apply cutinase, we degrade the cuticle, we release the DAMs, and when we apply the botrytis cinerea in the same place, then we have resistance. Another evidence is with the mutants, because several mutants, all of these mutants, Bodyguard, LAX2, Fieldhead, and Lacerata, they had been described, but very well described, to be mutants in the cutin-related signaling pathway and biosynthesis. These three mutants, they have these phenotypes. So all of them, they are resistant to botrytis. All of them produce more reactive oxygen species. We can detect it with this green color. without the pathogen. Yeah, see, constitutively, they induce the reactive oxygen species, and obviously, since they are not properly built because they are lacking from cutin, they have more leaf permeability. And this one you can see with these blue colors. So, how? Sorry, this is important because we characterize here in Arabidopsis, but it's not only in Arabidopsis where we can see. It's not for all the plants, but there are other plants, including tomato and also Camelina sativa. So in these ones, again, mutants in the cutin biosynthesis and transport, they have the same three phenotypes: resistant to botrytis, reactive oxygen species accumulation, and the permeability. So how we explain this resistance? It was a model that we made several years ago. So what we thought is that when we make holes or not proper constitution of the cuticle, they will be more permeable, as we can see, but they will be permeable for both sides, not only for the things that are coming from outside, but also for inside. And when they are more permeable, they will release and most likely detect more faster the dams and MAMs from botrytis cinerea. And this leads the plant the opportunity to build up the resistance mechanisms, all of these molecular changes, more faster. And that's why we have resistance. So that was our model. So more permeability, more reactive oxygen species, more innate immune responses, and resistance. Okay, this helped us to explain about the cutin changes. But then we realized that for the waxes, we didn't know it. So we never tried mutants related with waxes. So then that was the work of Wendy Aragon during her PhD to try to answer if change on the wax composition also leads to the resistance to botrytis cinerea. Fortunately, we work with Arabidopsis, and we have mutants for every combination. So we have the cutin mutants very well characterized, bodyguard. The wax mutants, SER1 and SER2, they were also characterized, not in relations to botrytis, but were characterized as that they have the changes in the cuticle, etc. And we have one mutant in our hands in the lab that is called ICA2. That actually, we describe it to be the first mutant that has cutin and wax, a reduction in the cutin and wax. So we have all the combinations, fortunately. So Wendy started to work with this, and then the first thing she did was, okay, let's check if there are permeable, all of these mutants. And so all the mutants were more permeable. We analyzed in different ways, yeah? And what you can see here, so we have in black, there are the wild types, because we have two different types of wild types. You will see this. C24 is the wild type of ECA2, and the cold series is the wild type for the rest. So as you can see here, for the cutin mutants, bodyguard and LAX2, they have more permeability, and SER1 also not as much as the cutin, but still there are more than the wild type, and for ECA2 also is more permeable. So check. This is the first check that we have for our model. All of them, they are more permeable. Then what happened with the ROS? Well, in the basal condition, as I say, the basal condition, normally the mutants, the cutin-related mutants have more ROS. What we observe is that actually also all the mutants have, not in the same level. Again, it was some level in the wax-related mutants, but still it was significantly more than the wild type. And of course, the cutin mutants, they have more ROS. So this is the basal. What happened when the pathogen is interaction with these mutants? Then you can see here more clearly that the mutants, all of them, they accumulate more ROS than the wild type. So again, it was more permeable, more ROS. So the next phenotype that we wanted to see or to analyze was the resistance against botrytis. And here, it was a very nice surprise, because only the mutants that we already know, the LAX and bodyguard and ICA, they were resistant to botrytis cinerea. But the wax mutants, they were not. They were as susceptible as the wild type. And I remember this, I tried this like an anecdote, because I remember when Wendy observed this, she didn't want to show me this. She was afraid that I was upset because the model was wrong. I say, no, come on, it's great, because then we can destroy our own model. So we can start all over again and to try to see it, because it was not enough that they were more permeable. They had more rust to have the resistance. And then we start to answer, okay, why the Q? And to do that, we start to collaborate. Well, we already collaborate all the time with Damien Formay, but they started to collaborate in this project. And what we decided to do, it was to try to answer this question using the transcriptomics. So we performed the transcriptomics for all of these mutants under the basal conditions and the infected conditions. I will not go through all the data, this is too much, but the most important results were that in the Q10 mutants, they induce the defense-related genes. And the wax mutant induced the abiotic stress-related genes. So it was a kind of logic, this response, and that's why in the Q10s, we have the defense against botrytis cinerea. Then we ask, okay, but which genes are involved in this defense? Because it was too general, yeah? And then we focus on the late defense responses, the genes related in the innate immune response and the late defense responses that had been massively described in the literature. We say, okay, probably those are the same set of genes, the canonical genes that are induced or repressed in the Q10 mutants, but not in the wax mutants. But then it was a nice surprise, because there's not really something here that we can say, okay, all of these 3 mutants that are resistant are yellow, all of them, and here blue, or vice versa. We cannot really see it. So it's not because the canonical defense responses are modified in the Q10-related mutants that might help us to explain the defense response. But then Wendy had to dip into the data and try to find these kind of genes. And unfortunately, she found it. She found several. Here's just a piece of the most important ones. And nicely, they was related to the cell wall remodeling. And of course, this was already not only with the RNA-seq, we checked it also with the qRT-PCR. And unfortunately, and also nicely, some of these genes, this PMT70, it was just 1 month earlier described to have a phenotype in the defense response against botrytis. So it was interesting. It was one of the first times that there was related this cell wall remodeling to induce the defense response to botrytis cinerea. So now we have a new model, yeah? Here for the Q10 mutants, so we know that they are more permeable. We know that they have more reactive oxygen species. They have induction of the defense-related gene expression, but also we have these genes that now we are trying to characterize related to the cell wall remodeling. And the wax mutants, well, they are more susceptible, even though they are more permeable, they still can have some ROS accumulation, not as much as Q10-related mutants, but the main difference is that they induce the genes related with the abiotic and photosynthesis-related genes. So with this, at least we move forward. We have new model to work on it, and hopefully in the future we can improve it or destroy it or whatever, yeah. And this was a work of Wendy. It was published already a couple of years ago. Okay, then we want to move forward and then to try something different. And this is to analyze the microtranscriptomic responses. Why? Well, here you know the microRNAs, or the small RNAs, sorry, they are involved in many different processes in the biology, not only for the plants, but also for other animals and microorganisms. Also, yesterday it was described that we have this cross-kingdom system where they are very, very involved in the small RNAs. Additionally, we make some reports. Last year, we have a report of the work of Emir Padilla. He made the master in my group, where we describe what happened. For the first time, we have a database of the small RNAs, during the interaction with botrytis cinerea on the early stage. The early stage, we mentioned 6 hours, okay? And what Emir found is that many small RNAs are induced during these early times. And just here, to highlight, it was interesting to find that many of the heterochromatic small RNAs are overrepresented. And that means that maybe the relax of the chromatin is important for the plant to defend against botrytis cinerea. And it's something that we want to pursue, but I will not go further than here. So also, we observed that many of the small RNAs had been already described to target genes that are related, and that is expected, with the hormones that are involved in the defense response of botrytis cinerea. So we know that the small RNAs are involved not only in the cross kingdom, but also in the defense response. Then we ask the question, okay, these small RNAs are also involved in the QT-related mutants, or defense response, sorry. And this is, again, the work of Carlos de la Rosa, who made a postdoc in my group. And we use our model that is a little bit more. ICA2 is a mutant with more cuticle permeability, more ROS. They have less wax and cutin. We know that they have primed the defense responses. And also something that is very interesting, because they is not only resistant to botrytis, but also to Pseudomonas and Spodoptera, but is susceptible to Phytophthora. So it's not that ICA2 is so stressed that that responds against everything, but is more or less specific for these pathogens. Okay, so what we did, it was to check the basal conditions, of the wild type and ICA2, and also once they are in interaction with botrytis cinerea 6 hours post-induction. What we found is the following. What happened? Okay, so Emir found that there are many small RNAs that actually are down-regulated in ICA2 under the basal condition, non-challenge condition. And again, he analyzed where they were distributed, these genes, and in this case, the stirnas were involved in this repression of the small RNAs. Then he focused to identify which small RNAs were either accumulated and then the target down-regulated, or vice versa. Small RNAs down accumulate and the target up-regulated. And he found many. I will not go of the pathogen-related genes that actually are down-regulated after the small RNAs are accumulated. Then we asked the question, what happened on the infected conditions? And here it was a nice surprise also that ICA2 have a really massive number of small RNAs accumulated under this condition at 6 hours. And again, it was induced, most of them, but also repressed. And he classified, again, here we see again the Myrnas and also the heterochromatic gene, heterochromatic related small RNAs. Okay, once more, he went through to the predictions of the pathogen-related targets, and he found many of them. Now the challenge is to see, okay, now what we do with that, which genes we select to characterize, to further characterize, and so on. It's something that we need students, if somebody is there, so we need the students to really try to take care about this. But to try to verify this in silico analysis, what we decided to do is to go to the literature, and we found several, not many, but several small RNAs that actually had been experimentally validate. And here I show you only one. This is the case of MIR773A, and the target of this gene. And you can see here when this is induced, when this repressed, the MIR, the small RNA, the target is upregulated, and vice versa. And also this one, the MET2, that has been already published to be involved in the defense response against pathogens. Okay, so now with this information, we can say that we have a new element that we can start to work on it. And this is the small RNAs in this model. And we wanted to go further to try to characterize. But as I say, we need money, and we need also students or postdocs that they can work on that. Okay, what else we are doing here at CCG related with this topic? Well, as I say, in the collection that Emir found, we have several siRNAs that are known to be transported in the vesicles. All of these, at least this one, we observed that they are accumulated during the interaction with botrytis cinerea. So we asked the question, okay, then it should be important the traffic of vesicles, or probably differentiate within the mutants that are important, this process. And this is the thesis of Norma Vilés, a PhD thesis. And then she wanted to characterize if the transport of the vesicles are involved in the defense response of the cutin-related mutants. And to do that, she isolated some vesicles after the collaboration with the group of Mario Arteaga. Also, we managed to improve the protocol to isolate the vesicles. And now the next step is to sequence these vesicles and to try to characterize one or two of these candidates during the interaction with botrytis cinerea. So far, other part of the project, so far I show you what we are doing with the bad relationships in the interaction with the plant and microbes. But also, we start to work also with the beneficial interactions, yeah? So, normally, during the interaction with the beneficial microorganisms, in this case, this is just example of the mycorrhiza, well, we have the MIC factors that are recognized. They induce the common symbiotic pathway, and then the hormone signaling pathway is induced that leads to the mycorrhization. In the fungal pathogen, I already showed you that they are induction of the late defense response before the innate immune response of the ROS. But the important thing here is that we know, or we expected, that the small RNAs are also involved. And the question is, the main question is really, what are the small RNAs that the plant can use either to stop the fungal pathogen or to induce the mycorrhization of, to enhance these beneficial interactions. And to do that, what we are doing is to analyze one microorganism that is Serendipity indica, because this is a mycorrhiza, and again, botrytis cinerea, in the interaction with Arabidopsis. Serendipity indica, is a well-characterized mycorrhiza that improves the health of the plant. Yeah, it helps to manage to avoid the biotic and abiotic stresses. And very interesting, they interact with many different plants, mono and dicotyledons. And the most important for us is that they can interact with Arabidopsis. It's the first time that one organism has been really characterized that interact with Arabidopsis. And we already know, they have been described, how is the process, infection process of with Arabidopsis. And the important thing in the end also is that we can answer why for the plants, we already see many of the talks. In the legumes, we have the symbiotic pathway, all of these genetic elements. For the monocots, we know that they can interact with the mycorrhiza, but with botrytis, we still don't know. So it can open the opportunity to try to identify how Arabidopsis can interact with the mycorrhiza and to establish these beneficial interactions, and how are different compared with these other two groups of plants. So to do that, this is a work from the group of Damian. This is a close collaboration with him, and was made by his student, Ana Karen Sandoval. And what we did, it was, okay, we know exactly the times where S. indica interact with botrytis. So we select that time points to have the samples to perform the experiment for each time point. And then we make RNA-seq, we analyze also, we sequence the small RNAs, and we date the degradome. So I will not go into the detail, but we found many small RNAs that actually are induced during this interaction and at different times, 136. And this is, we are already finished, almost finished the paper, and we will submit it soon. But the important thing is now we can compare, because we have our own data, we have the data from the S. indica, and now we can see what are the genes that they share between these two different interactions. And this is something that we are working and we are pursuing, and we are very interested to continue this analysis. Okay, finally, another work in the collaboration that we have. Well, Mario already showed all the results. I will not go through. With Anna and Mario, we have this collaboration with Mercantia, also with the cuticular mutants in Mercantia, that one day we will show you, or probably Mario will show in the future, the results. Also, we have another collaboration with Claudia Martinez, the former PhD student in her group, Delia, and also again, Wendy. And this is working with the Pectobacterium. This is a bacteria that is also necrotrophic as botrytis. And here we apply the same, we ask the question, okay, with the fungus, we know the necrotroph are resistant for the cuticular mutants, but what happened here? Well, it was exactly the contrary. So the cutin-related mutants, they were susceptible, but the wax, they were more resistant. So now we are trying to further characterize this. We have already, we wanted to publish this result. This is only one of the part of the results. But what it tells us is that really the plant microinteract, we already know. So they are very complex because we have one phenotype and one clear phenotype within one pathogen, but exactly the opposite using another pathogen. So with that, I wanted to thank the agencies to give us some money. And also this is teamwork. And I wanted to thank all the people who has been involved, my group. Yeah, the latest picture of my group, and other people I already mentioned has been involved, but now they are not longer in the group. And also I wanted to thank the collaborations of many institutes in Mexico and over the outside Mexico. And of course, they are always welcome, this collaboration. I'm looking forward for this collaboration. Hopefully we will have more after this meeting. And finally, I just want to thank you. And here you can contact me. I'm looking forward for your questions.
Thank you, Mario. Yeah. Any questions in the audience? Laila?
Thank you, Mario, for your presentation. I was wondering if these wax cuticle mutants, you saw that more abiotic responses. Do you know if have you tested if they really are more resistant to abiotic stresses per se? Like, for example, I don't know, radiation or salt or drought?
You know, what they have been characterized, not only the wax mutant, but also the cutin mutants, is an abiotic stress-related phenotype. So they lost more water, as expected, because they are more permeable, they lost more water. And yes, so they have generally more susceptible for the abiotic stress, yeah, in general. Also, there are other pathogens that modify the interaction in these mutants, but I don't remember exactly the details, so I don't want to tell you some lies. But yes, they have been also described.
And in the case of serendipia, well, it's a really like mycorrhization, and you see arbuscules and everything, and also they get phosphorus for the plant. It's already also established everything.
Yes,
Okay.
it's well established there.
Okay,
Yeah.
thank you.
I had many questions, and then you mentioned that you showed the results of how the mutants behave differently, and everything was destroyed in my mind. But I have one. So Q10 mutants have more permeability, right? So would you expect them to have more vesicle transport? Like they are more capable of just throwing everything?
That's what we expect. That is the hypothesis. So Norma is working on that, and yeah, this is in the end. This is the hypothesis that since they are more permeable, probably they diffuse faster or more. We don't know. That's why we need to count if we have more vesicles moving around, or if the content is different. So there are two things. Okay. We expected that they are more efficient and more vesicles moving around because the number of the small RNAs that after interaction with botrytis, but we don't know yet.
Okay, yeah, because it sounds to me like some sort of vaccine, the Q10 mutant, like it's already primed to respond, but then that doesn't fit the results of why it's not working with other pathogens. But I don't know enough to, maybe it's obvious, but just to me, it's very surprising.
Yeah, I mean, one might expect that if they are more resistant to different pathogens, they could be resistant for all the pathogens, but it's not the case. That's why in ICA2 at least, we know that it's more resistant to Pseudomonas, this is a bacteria, to botrytis, and also to herbivore, but is more susceptible to Phytophthora. So I think my idea is that it depends how the pathogen interact with the plant because they don't use the same mechanism. So, and it was okay. It was nice because I mean, one might expect that the plant is so stressed that then can be just to mess up that they respond to everything, but it's not. And you can see the plant, they have a phenotype, so they have a reduction about 20% of the size, but it's not so stressed that you can see easily. Also, one readout that we can say, okay, yes, is prime, but not fully prime, not fully induce a different response, is with the ROS. They have a basal level of ROS, but still, once the pathogen arrives, it goes really further.
Okay. And can I ask one more question? And the results you showed of EMIR, of the small RNAs, where you identify them without infection and with infection. Without infection, if I understood correctly, you see more small RNAs downregulated. And then you infect and you see more upregulated. Is that relevant?
No, both. Sorry, I messed up with that. So in the basal conditions, yes, they're more downregulated. But once is with the pathogen, they have more or less similar number, but down and up regulated.
Okay, so it's not so stressed.
If it's relevant, I cannot answer that yet. Because, I mean, we observe that in the basal condition, they have many genes related with the different response that are induced or they are induced because the small RNAs are downregulated and they are induced, the targets. But still, it has to be verified, experimentally verified.
Thank you very much.
Luis?
Thank you, Mario. I have two questions. The first one is related to the cuticle mutant. And you were saying that, the pectin methylesteroids gets upregulated. That's very interesting. So do you think that in the cuticle mutants, probably, well, the plant cell wall is somehow more exposed, and these pectin methylesteroids could be able to desterify the pectin and then to get it more rigid with binding with calcium? Or what is your idea about that?
My idea is that yes. So in the short, yes, it should be modified. We don't know exactly at which level, and that's something that we have to really investigate because we don't know exactly. So we identified these genes, these cell wall related genes, but we don't know yet. We haven't really checked the phenotype related to the cell wall.
But
We didn't.
it would be nice to see the subcellular localization and see if that increase in the cell wall in those mutants compared to the
Yeah,
to
we
the
will
wild
do
type.
it.
And my other question is related to the ROS productions. As Daniela was saying, well, that there is this priming response because you have like higher reactive oxygen species. I wonder if these cuticle mutants also have this priming in the root. How do you look at the root and see if that is like a general response that you could have like increased ROS levels?
Look, for the roots, well, first of all, in the roots, there is not cuticle per se. It's not cutie, nor waxes. They are suberin.
Right.
Yeah. They have a very similar structure, and also the biosynthesis is similar. And they have been only described the cutin on the tips of the roots.
Exactly.
That is also important because there is where the microbes interact a lot.
Right. Mm-hmm.
So we haven't checked really. I was just focused on the upper part, but we wanted to check with these mutants with interaction with the Simons, also with the S. indica,
Right.
Serendipity indica, with the mutants to see if they have difference. But we really haven't really managed to do that. So this is the first thing. And the ROS, we haven't checked in the roots, to be honest.
Yeah.
So we don't know if it's a general thing that
like
they accumulate the
systemic,
ROS or systemic, or if it's really localized on the upper part of the plant.
Okay.
Don't know.
Yeah. So is Magnaporte also like a pathogen for Arabidopsis? Magnaporte.
Yeah.
Because
Yeah,
it's very interesting because, as you said previously, some of the pathogens requires to stop the reactive oxygen species, but some others like Magnaporte, they requires to produce the reactive oxygen species so that they could be effective. So yeah.
No, I don't know, to be honest. I don't know
right.
if they have.
Thanks.
Thank you, Mario. Very nice data. Just a few questions. The first one is related to the phenotype of the cuticle mutants. Did you see any change in the growth under standard growth conditions? This is the first one. And another related to this question is, did you see any change in the growth of the root in the Petri plate assay?
For the upper part in the growth for the cutin mutants, they have many. So it depends which mutant we are talking about. For instance, in the bodyguard, they are very, very messy plant because they cannot, since bodyguard is related in the transport of the cutin, is really a very important step. They have fused the organs, yeah? So they have a really strong phenotype. For ICA2, as I say, they have a reduction of the size of about 20 to 50% of the reduction of the size in the rosette, in the upper part. So in short, yes, they have a phenotype. They have a normally reduced size in the upper part. And for the roots, to be honest, we haven't focused on the roots. I don't know, to be honest. But as far as I remember, well, I check the plants once we are growing on the in vitro conditions, and they grow normal, let's say, for the root, but I cannot be 100% sure.
The possible link is the perception of UV light in leaves. Yeah. And in addition, that there is an increase in rows as you showed us. So this is a very strong stress in the shoot system. Yes. And this stress is perceived in the shoot in the root meristem. So there will be a direct effect in the activity of the root meristem. Yeah. This was my curiosity about these results.
Yeah, we haven't checked really because we were really focused on the abiotic interactions, but yeah, sure. For sure, it will be interesting to analyze.
Well, the last one is, did you see, do you really see the proliferation of the serendipita in the Arabidopsis roots? Because Arabidopsis is not good, it's not a good model for mycorrhiza colonization research.
Yeah, I know.
It's hard to work with. The root is very, very thin.
Yeah,
And there is no good recognition between beneficial fungi and this model plant. So this is my question: Have you tested the proliferation of the root inside the proliferation of the fungus in
Inside.
the root system?
yes, indeed. Not only also it was already published when they described the infection process. They did a very nice microscopy and they can find the bodies inside, the mycorrhiza inside, kind of mycorrhiza inside the roots, but also in our hands, we can see not so easily, as you say, it's not the best model, but I mean, it's a nice model because then we can analyze and we can take all the advantage of Arabidopsis.
Yes.
Yeah, but yeah, it's not, it happens, so yes, it interacts. In some aspects, it's not the normal mycorrhization, yeah, but yeah, interacts, yeah.
Okay, may I have the last one? Well, this is just a curiosity. Why do you focus on the small RNAs as messengers? Why not to test directly the transcriptional responses following your favorite treatment? Just to apply the treatment, check your mutants, change in the phenotype, and then check the transcription factors, receptors that change the expression.
Okay.
Why to include this pathway of the small RNAs?
I think for two things. First one is because we already have the RNA-seq analysis, so we already know. And for the small RNAs, because we like to do it. So it's, we have an expert at home. So with Damian Formey as an expert. So I think it's important and it's an interesting piece of information because then we can add a little bit, a small layer of regulation by these molecules related to the transcription that we already know. So that's why it's really driven by the curiosity, basically.
Okay. Thank you.
And also, let me tell you also, so the small RNAs are a very potent biotechnological weapon because it's already known that these small RNAs can be applied directly to the pathogens and inhibit the growth, or even to induce the defense responses. So behind the basic research, if we can find something that we can apply on the field, the small RNAs on the field, then it would be nice. But the main question is because I'm curious. That's it.
Thank you, Mario.
Yeah, thank you, Jose.
I have a few questions, but I try to keep them short before coffee.
Yeah.
So you see that in some of your cuticle mutant, you see constitutive ROS production, and then you see this priming effect. The cuticle can be a source of elicitors. So do you think that in addition to what you propose, that you have maybe increased permeability of vesicle, that you basically release elicitors, and that's why you can't have constant activation of immunity?
This is a very interesting question. Thank you, Cyril. Actually, what happens in the QT mutants is that it's an overproduction of the wax. When they cannot produce Q10, they overproduce wax. And for sure, this overproduction is like a dam, and when they are not properly built on the cuticle, again, there are dams, and that's why we have the ROS. The basal level of ROS is increased. And of course, it can be also triggering the defense response indirectly. Yeah.
So that's a link to my second question. So you see this ROS, and as was mentioned before, this could lead to this growth inhibition that you observe. So did you test if you make a double mutant with RBOHD, for example, that will block apoplastic ROS production, if you restore normal growth?
We haven't tested. We haven't did it.
Because this will provide a causal link between the two phenotypes.
Yes, that's true. And also we can rule out if the ROS really is important for the phenotype
And my third point is just to clarify something. You have now this collaboration with Damien where you compare the infection with serendipita and botrytis, and you want to see commonalities and differences. In this case, are you infecting the root as well with botrytis? Because otherwise you compare leaf with roots.
Yeah, that's true. But you know, yes, this is one of the downhill, is not so clean experiment, because you're right. For serendipita, we infect the roots, and for botrytis, we infect the leaves. Even though we extract the tissue, the whole tissue, roots and leaves, yes, we will find something that is only expressed or accumulated on the leaves, and vice versa, only on the roots. But we have to try it, to test it, because one idea is, once we found this link or this common accumulated or differentially accumulated small RNAs, we will have the mutants or overexpressing, and then we check in both systems, in those pathosystems. Yeah.
Thank you.
Yeah, thank you, Silvio.
On the last
Mario,
question, please.
yes.
The last, I think, because to keep
Over here.
Thank
Hi,
you.
Mario. I just have a question about I'm curious about how you inoculate the leaves. I mean, is it through infiltration or how? But I wonder, I mean, at least we know a little bit about the infection process of Pectobacterium. And we think that it's necessary a kind of swarming and this kind of mobility. So if there's not because of this infection protocol, the proliferation, and I mean, from the perspective of the microbe, there is it is not working. So because these mutants, the physical properties of the surface are modified, no? I mean, so I'm curious about the inoculation process. I mean, because maybe from the perspective of the micro, it is what is happening, no?
No, it's not. The microorganism is not forced to go inside. We just put it in the droplet on the top of the leaf and leave it like that. Yeah, you can drop it or you can spray it over the whole leaf. And then it goes, normally botrytis cinerea start to sense if there is on the leaf, they germinate, and as yesterday Mario showed, they start to interact with the leaf without any external force. So it's not like Pseudomonas that you really have to infiltrate, like Pectobacterium that you have to infiltrate. No, you just put it on the surface and then they go alone.
Okay, and this Ross species is also what I think you mentioned a little bit that is it's like well, it's more sensible to the environment, and it's why it is like this Ross species activate. So what is the explanation of this Ross species? So it is because it is sensing like it's more sensible to the environment, or
well, it's one of the explanation is what Cyril says, that the defect of the cutin acts as to produce dams, and then the dams are known to induce the reactive oxygen species accumulation. This is one. The other one is, as Jose said, that the light can be also, since they don't have the
accepted to come and give a talk. And he will be closing the symposium. So thank you, Osvaldo, for coming. And he's coming from FES Iztacala. And his talk is about the understanding of the genetic control of root nodule symbiosis under phosphate deficient conditions. So thank you, Osvaldo. Go ahead.
Presenting our data. So, but also I'm really happy to be back here to my alma mater. I did my PhD here 20 years ago. Exactly 20 years ago, I started my PhD. And since then, I have this question in my mind that how phosphate levels control the root nodule symbiosis. And I have to admit that there is many retractors. So now we are on the review in our manuscript, and they say, no, I mean, phosphate is not really important for the root nodule symbiosis. But we have different evidence demonstrating that, yeah, it's not only a mineral nutrient. It is really important to shape the symbiosis with rhizobia, but also we know that phosphate is really, really important to shape the interaction with different microbes, either pathogens or also beneficial microbes. But they say, well, nitrogen is the most important and other nutrients are not really, really relevant. So that's why today the title of my talk is Toward the Understanding of the Genetic Control of Root Node Symbiosis under Phosphate Deficient Condition. So here, this picture, I will present more details about that, but I just want to bring your attention that this is optimal phosphate condition, this is low phosphate condition, and this is the master regulator of the root nodule symbiosis, which is nodule inception. So you can see that there is a huge impact in the expression of NIM. And this one, this is another master regulator, but the phosphate deficiency response. So this is PHR1, this is a transition factor, and this is under optimal phosphate condition, and this is under low phosphate condition. So you can see the contrast response of these two master regulators. So I'm really lucky that Luis, Gina Hernandez, Jesus already talked about root nodule symbiosis, so I can skip many slides of introduction. I just want to bring your attention that root nodule symbiosis is a really important process to feed the food chain with fixed nitrogen. So this means that if this symbiosis fails, so basically life in the air will be untreated. So that's why it's very important. And I got, I would say a few years back, so a very nasty email like, why you're keeping studying root nodule symbiosis? So there is no more new about this topic. And I say, well, this is not completely true. There is many aspects. And one of these aspects is how nutrient availability regulates this symbiosis. So it's not only nitrogen, as I say at the beginning, there are other nutrients like phosphate, like iron, that are really, really important to sustain this symbiosis. So that's why I keep researching on this topic for several years. So as I mentioned, so I don't want to, I mean, no, I don't want, no. I will not go in details about all this analyzation, but I just want to bring your attention that since 20 years ago, the community has spent a lot of time and resources to understand all the communication between legumes and rhizobia. Now we have a big picture of how rhizobia communicate with legumes. We know basically, I will not say all, but I will say the majority of the genetic programs required for rhizobial infection, nodule development, and also nodule functioning. So I will not go in detail as I say, but I want to bring your attention that our research group make well, to me, a significant contribution because, so we know basically what happened when the rhizobia communicates with legumes. And we know that after node factor perception, different genetic programs will be activated to form a nodule. But there is a huge gap, what is happening when rhizobia is perceived and what happened after the plasma membrane to the nuclei. As Luis mentioned, that the calcium spiking is really important to sustain not only this symbiosis, but also the symbiosis with mycorrhizae. So a few years ago, when I will not say few, I say like 10 years ago when I was postdoc with Jean-Michel Anet, so we discovered this gene, early phosphorylated protein 1, because after 30 minutes, this protein got really phosphorylated. So the question was, what EPP1 is doing in this symbiosis with rhizobia? So I will not go in details, but I just want to mention that when we silence EPP1 in Medicago truncatula, so we observe that there is no calcium spike. So it indicates that EPP1 is required to activate the calcium spike. So you can say, yeah, but this is transgenic roots. So we validate this with stable transgenic lines, and we got exactly the same results. So EPP1 is required to activate calcium spiking. So we know that serine 77 is phosphorylated. So what we did is we overexpress EPP1, the wild type version, But also we generated different phosphomutants, the phosphoactive mutant. In this case, I'm showing the data. But the most interesting part is when we overexpress EPP1 wild type or the phosphoactive version, so we were able to activate part of the common symbiotic pathway. So this means that EPP1 alone can activate the molecular dialogue between legumes and rhizobia. So the most interesting part is that when we overexpress EPP1 in the background, in the mutant background DMI2, so we reestablish the symbiotic phenotype. So this means that EPP1 is acting just downstream of DMI2. So also we did RNA-seq analysis, and we observed that the majority of the genes involved in rhizobial infection are upregulated in the absence of rhizobia, just by overexpressing wild-type version or the phosphomutant version. So this also supports that EPP1 is really required to activate the common symbiotic pathway. So we came out with this model that EPP1 is phosphorylated here in this manuscript that is published in New Phytologist last year. So we say this is an unknown protein kinase, but now we have evidence that DMI2 or CMARCate is phosphorylated EPP1. And in some way, we are proposing that EPP1 is contributed to the biosynthesis of secondary messengers. So very likely, mevalonate. And we know based on available data that mevalonate is required to activate the calcium spike. So EPP1 is very likely the link between the plasma membrane and the nuclei, and it's really important to sustain not only root node symbiosis, but also the symbiosis with arbuscular mycorrhizae fungi. So basically, what we found here is a new member of the common symbiotic pathway. And collaborating with Jean-Michel and Pierre-Marc from France, now we have more data in Mercantia, indicated that EPP1 is required to synthesize different secondary messengers. I will not tell you the names of these secondary messengers because now it's sort of top secret, because through the evolution of the land plants, EPP1 is playing a huge role in sustaining different endosymbiosis, not only root nodule symbiosis or AM fungi symbiosis, other type of symbiosis. So this is the task that we have as researchers to understand how legumes are communicated with rhizobia. So we made a little contribution to this research field. But over these 20 years, and I will say more than 40 years of investigation, now we know that there are different peptides required to activate the symbiosis with rhizobia. We know both negative and positive regulators. So in the case of positive, under low phosphate conditions, there is two peptides, CEP1 that is very likely activated by low nitrogen condition. CEP7 is activated by low nitrogen condition, but also by rhizobia. And these peptides travel to the shoot where CRA2 receptor will detect it and will promote the accumulation of this microRNA 2111 to make susceptible the plant to be colonized by rhizobia. So this is, in summary, the activation of the symbiosis with rhizobia. And I just want to mention that very recently, so there is a paper indicated that these two peptides that are activated by low nitrogen or rhizobia, they're also activated by low phosphate conditions. And the question is, why low phosphate is activating two positive peptides for the root node symbiosis? But the very interesting part is that if there is low phosphate condition, these two guys will inhibit the interaction with rhizobia and will promote the symbiosis with AM fungi. So is phosphate important for the root node symbiosis? I think we have more evidence to say, let's say very likely. I will not say yes, because the detractors, let's call it like that, will say, yeah, you don't have all the evidence, but yes, there is evidence. And here we have the negative guys. So there is the clear peptides that under abundant or optimal nitrogen condition, they will be produced and they will restrict the symbiosis with rhizobia. Why? Because this symbiosis is really energetically costly. So here we have some numbers that it requires 16 ATPs, but this is only for the nitrogenous activity. If we take in account when the nitrogen complex is assembled, so there is more ATPs, it's about 32 or 35 ATPs. And this number is only for one single molecule of atmospheric nitrogen. So it's really costly. But the plant have evolved a signaling pathway to control and avoid metabolic imbalance and sustain the symbiotic nitrogen fixation, but also to sustain plant development. So I will go back in a few slides with this model, but I just want to bring your attention that this model is made on soybean. So it is called rhizobia-induced cleptide 1 and 2. So RIG1 and RIG2 will travel again to the shoot where NARC receptor will perceive and will induce. Some people say now we know the chemical identity of this signal molecule, but there is controversy. So let's call an shoot-derived molecule that will inhibit the interaction with rhizobia. So, and one of the most important players is too much love. Too much love, even we don't know what is, this is a G3 ubiquitinase ligase. So in theory, it will target NIM to stop the nodule formation. But there is no contundent evidence supporting that TML is targeting NIM. But the hypothesis mentioned that very likely NIM will be targeted by too much love and stop the symbiosis. So more than 40 years ago, so Peter Greshko from Australia came with the idea that what happens if we have a supernodulation plants? So can we have a plant that can self-fertilize it? And we have a super plant. So it wasn't the case, because here you can see the wild type forming exactly the right number of nodules. You can see the size of this plant. In contrast, when we have a supernodulated plant. So we have a smaller plant and even is struggle to grow. So this confirmed that the nitrogen fixation process for legumes is really, really costly. So, and that's why the plant needs to control the number of nodules. And now we know also plants control the number of rhizobial infections. As I mentioned, for plants it's more cheaper to obtain nitrogen from the soil as nitrate. So when the plant is growing in soil with optimal nitrate condition, it will stop the interaction with rhizobia. But it will be promoted when nitrate is not available or is in lower concentrations. But now the question is there another mineral nutrient that regulates the establishment of the root nodule symbiosis? And of course, you know, so my favorite one is phosphorus, but there is a very recent paper talking about the importance of iron in controlling the establishment of this symbiosis. So today I will not talk about iron, but I will focus on phosphate. And as you know, phosphorus is the second most important mineral nutrient for living organisms, not only for plants, but for all the organisms, because it is required to synthesize DNA or RNA, to synthesize phospholipids, but also is important for signal transduction through phosphorylation and dephosphorylation cascades. And if we look very carefully in the signal transduction, in this symbiosis, there is different phosphorylation steps that are crucial to engage in symbiosis with rhizobia. So it is important, phosphorus, to sustain this symbiosis. So the problem with phosphate it is that phosphate, it is really reactive element because plants only can take phosphorus from the soil as phosphate, but phosphate rapidly interact with different cations, making insoluble precipitates that the plants cannot uptake. So basically the plants all the time, are dealing with phosphate deficiency. And phosphate deficiency is an international problem because most of the arable soils experiment with some level of phosphate deficiency. If you think about that, legumes are growing in some of those arable soils, basically the nitrogen fixation process, it is affected or compromised just because the phosphate availability. And I know that there is an international consortium working or making possible that non-legumes can fix nitrogen in association with rhizobia. And in my opinion, if we don't take in consideration the effect of phosphate status of the plant, so this bigger effort will be also intrigued. Because not only legumes are dealing with phosphate deficiency, all the plants are dealing with that. So we need to put more attention in the phosphate availability. So phosphorus is important for, obviously, nitrogen fixation, but also basically it is important for each step of the plant development and plant growth. And this includes legumes, not only model plants or not only other crop plants, legumes are also affected by phosphorus. So here you can see the negative impacts of phosphate deficiency. So here we have plants growing under optimal phosphate versus low phosphate. So it is really affecting the plant growth and also plant yield. And you can see here how the plants modify root architecture to deal with phosphate deficiency. So thanks to Arabidopsis, I was listening the talk from Jesus Lopez Bucio that I say, yeah, but it's Arabidopsis. Yeah, but thanks to Arabidopsis, now we know most of the genetic players that controls the response, the plant response to phosphate deficiency. So I have to admit, thanks to Arabidopsis, and we can translate this knowledge into crop plants like legumes. But I just want to bring your attention to this master regulator that I mentioned at the beginning of my talk, which is PHR1. This transcription factor will recognize or will bind to this cis regulatory element that we call PHR binding sites, or P1BS. So the majority of the phosphate deficiency responsive genes will have at least one of these cis elements in their promoter region. So some examples are phosphate transporter, phosphate translocators, so that are very important to sustain phosphate transport and also phosphate homeostasis. So PHR, it is an important guy controlling the plant response to phosphate deficiency. So how PHR works? So now we know, based in different studies, that the phosphoinositol pyrophosphate molecules binds to these SPX proteins. Under optimal phosphate condition, the pyrophosphate interaction with SPX protein will interact with PHR, and this will avoid that PHR activate all the responses to phosphate deficiency. So under low phosphate condition, so the interaction of SPX proteins with inositol pyrophosphate will be not longer sustained. And this will allow PHR to move to the nuclei and activate all the machinery required to face phosphate deficiency. But also, just almost 4 years back, so there is a beautiful paper talking about that PHR, it is an important player to sustain the symbiosis with arbuscular mycorrhizae fungi. But the most important, or the most interesting, at least from my perspective, is that PHR is required to activate CMARK or DMI2. And also, if we go back a little bit, so here is Cyclops, And all the guys working on these two symbiosis, we know that Cyclops is really important for the common symbiotic pathway. So the question is, does PHR controls part of the common symbiotic pathway based on the phosphate status of the plant? It is PHR controlling or deciding which microbe partner is the most appropriate to fight with environmental conditions. So in the case of the root nodule symbiosis, so this work is from the Jean Hernandez group, long time ago when I was a PhD student. So we noticed that under low phosphate condition, the number of nodules are significantly reduced, more than 50% reduction. But not only the number of nodules are affected, also the nitrogenous activity is significantly reduced. So phosphate has a negative impact in forming the nodules, but also in the functioning of those specialized organs. So there is a lot of effort to understand how nodules adapt to phosphate deficiency. So there is many papers, there are some examples, but this paper indicate that nodules adapt to phosphate deficiency by modifying the phosphate transport and telling the rest of the plant, you need to send me all the phosphate that you have to sustain the symbiosis and to sustain the nitrogen fixation. But the other organs say, why do I have to send you all my phosphate? I need to fill my seeds out. But this is the high demand of phosphate. So we know a lot of metabolic and transcriptional response of the nodule. But the main question is how phosphate is affecting the establishment of this symbiosis. If there is a genetic machinery involved in controlling the symbiosis under low phosphate condition, or is just a change in the carbon phosphate ratio, and there is nothing related to genetics. So this is the question that, since I was PhD students, I want to answer, but when I did my postdoc, I did in plant pathogen interaction. After that, I decided to go back to the plant symbiosis interaction. So when I moved to Mexico, I just start to try to answer all of these questions, like how does phosphate deficiency affect the root nodule symbiosis? Does phosphate deficiency affect the rhizobial infection process? Is the autoregulation of number of nodule pathway involved? Does PHR participate in the control of the root nodule symbiosis under phosphate deficient condition? So I have spent like almost 12 years working in the lab trying to answer this question. And I have to admit, Jesus, I'm a Physiolus vulgaris supporter. But I have good news for you at the end. So the first question is whether or not phosphate deficiency affects rhizobial infection process. Here we develop rhizobia expressing a marker gene, which is UA or GOS. So here we have roots infected with rhizobia under optimal phosphate condition or low phosphate condition. And you can see how rhizobia is attached to the responding zone. And the intense of the color indicates also that the successful of this interaction. But in low phosphate, you can see that even this responding zone is moving close to the root meristem, but also the intensity of the attached rhizobia also decreases. So here we have an evidence indicating or suggesting that the rhizobial infection, it is affected by low phosphate. So what we did is to analyze the expression of key symbiotic genes, which is NSP2, participated in this rhizobial infection process, but also participated in activating the expression of NIN. So obviously, we tested also the expression of NIN and floating-in-2 involved primarily in the rhizobial infection, but in the curling of the root hair. But in all cases, we observed that the expression of these symbiotic genes are reduced under low phosphate condition. So there is another evidence that phosphate status is at least reducing the expression of key symbiotic genes. So we generated a promoter fusion with GOS just to check if the spatiotemporal transcriptional activity of NIN is affected by low phosphate or not. So here we have optimal phosphate condition, low phosphate condition. This is Phaseolus vulgaris roots. And you can see here, in response to rhizobia, these patchy patterns starting from the epidermical cells, moving to the cortical cells, but also we can observe this transitional activity in root hairs and also in some that looks like are correlated by rhizobia. But in low phosphate condition, this patchy pattern and even the activity in root hairs is not longer observed. So low phosphate is not only reducing the expression of NIN, it's also affected the spatiotemporal activity of this master regulator of the root nodule symbiosis. So now we want to know if the expression of NIN and also this negative regulator that I explained earlier, which is too much love, and if PHR is the master regulator of the plant responses to phosphate deficiency, we wanted to know if this transitional factor, it is affected or not. So as you can see here, we have optimal phosphate condition, we have here empty vector, NIN, and here we only fused the small part of the promoter containing the binding site for Cyclops, which also participate in the activation of NIN. So you can see here that NIN requires optimal phosphate condition to activate the expression. If we compare this abundance of this transcript or the activity of the promoter, you can see that under low phosphate condition, it is significantly reduced, and in particularly, the promoter region containing the cyclone binding site, basically there is no signal. And we observed the opposite behavior for too much love, that low phosphate really increased the transcriptional activity of too much love promoter. And similarly for PHR. And this is very interesting because in the first round of review, the reviewers say, well, GOS staining is not enough to conclude this. Please analyze the expression level of GOS. So we did it. So basically it matched with the GOS activity by staining. So the transcript level of the GOS gene basically matched. We returned the manuscript and the other reviews say, well, what if the endogenous gene is not behaving like the promoter region that you fused or you used it? So we did qPCR analysis, and you can see here for NIN. So 5 days after inoculation, there is a significant reduction in the transcript levels. And you can see that this behavior is basically the same across different times of the nodule formation, 10 days of inoculation, 15 days, and 21. So basically it matched what we observed. But not only NIN, TML also have a positive response to phosphate deficiency, similarly for PHR. So here we can conclude that low phosphate is affecting the expression of NIN, but promoting the expression of negative regulator like TML. And we don't know what is the effect of increasing the expression of PHR. So as I mentioned before, PHR binds to P1BS cis regulatory elements. And if we are observing that this symbiotic genes has a response to phosphate deficiency, we want them to know if the cis regulatory elements P1BS are present or not in the promoter region. And very surprisingly, so NIN in Phaseolus vulgaris has 2 P1BS regulatory elements. Soybean has 4. As you know, soybean has at least 4 copies of each gene just because the duplication events that experimented this legume, Medica truncatula has 2 and Lotus has only 1. Similarly for TML. So this suggests that maybe PHR are controlling or modulating the expression of these 2 symbiotic genes. So what we did was an EMSA assays, and basically what we observed is that PHR binds to the promoter region of NIN. So this is another evidence that suggesting that PHR may have a role in controlling the expression of these 2 symbiotic genes. So what we did was a transactivation assays in Nicotiana ventamiana, and it was a mistake because we grow our Nicotiana ventamiana under low phosphate condition, but in the presence of nitrogen. But even though PHR transactivate the expression of NIN and TML, and here you have the quantitative data based on qPCR. And this is the other point that the reviewers are complaining. So wait. So you are telling me that NIN that only responds to rhizobia. You're telling me that low phosphate is activated, and I say yes. But why? For over 20 years, all the literature indicate that NIN is only participated for root nodule symbiosis. I say, well, so what I did is to do transactivation assays in transgenic roots of Phaseolus vulgaris, and basically, again, so optimal nitrogen condition, low phosphate condition, and also in roots, we can observe that PHR transactivate NIN. And here, TML has a very lower expression in the absence of PHR, but when we overexpress PHR together with the transcriptional fusion of the TML promoter, we can see an increase in the transcriptional activity of these promoters. Again, this is under low phosphate condition in the absence of rhizobia. So low phosphate under low phosphate condition and in the absence of rhizobia is activating this master regulator. So what is the role of NIN under a non-symbiotic scenario? So this is the question that we are working now in the laboratory. So we did the same transactivation assay, but in the presence of rhizobia and the absence of, in the absence of rhizobia and in low phosphate condition. And we observed the opposite response. When we overexpress PHR, we observe a significantly reduction of the activity of NIN and TML. So this data suggests that also the transcriptional activity of PHR has to be highly regulated. Not only overexpression will have a positive impact. We are observing that it having a negative impact. This suggests also that the spatiotemporal activity of PHR has to be regulated to properly activate or regulate the expression of symbiotic genes. So we did an ovulation assays in transgenic roots expressing the PHR RNAi lines or overexpressing. So here we observe that when we silence PHR, so there is a significantly reduction of the number of nodules in both conditions, in optimal phosphate or low phosphate condition. But when we overexpress, no, sorry, this is the transcript levels. Yeah. So here, this is the nodulation assays. So you can observe here that under low phosphate condition, there is a significant reduction, which is in line with previous reports. But when we silence RNAi, we no longer observe the negative impact of low phosphate in the number of nodules. Basically, it will form about the same numbers that the wild-type roots forms in optimal phosphate condition. But when we overexpress PHR, there is a significant reduction. It doesn't matter if there is phosphate or not. There is a significant reduction. And again, this data also confirms that PHR has to be tightly regulated to control the number of nodules based on the phosphate status of the host plant. So we did an RNA-seq analysis, again, in RNAi or overexpressing PHR roots. And basically, the summary of this figure is that different genes involved in the organogenesis of the nodules, but also in the autoregulation of nodulation, are affected by just overexpressing or silencing PHR. So this data also add evidence that PHR, it is very likely another genetic component to regulate the root nodule symbiosis under different phosphate conditions. So with this data at this point, we can conclude that phosphate deficiency reduces the expression of crucial symbiotic genes. Phosphate deficiency affects the spatiotemporal transcriptional activity of NIN, and phosphate deficiency upregulates the expression of NIN and TML in the absence of rhizobia. This is, for us, the most interesting data, because now we want to answer why NIN it is important for the plant under low phosphate condition. It is required to properly adapt to phosphate deficiency condition. It is required for AM fungi symbiosis, or what NIN is doing under this particular condition. And PHR is a fundamental genetic element in the establishment of the root node symbiosis. But now the other question is the autoregulation of nodulation pathway involved in this process? What components of this pathway are required to regulate the number of nodules under low phosphate condition? So what we did is we are really lucky to have a Phaseolus vulgaris mutant plant for the NARC receptor, and we use it to answer if the autoregulation of nodulation pathway is participated or not in controlling the number of nodules. So what we did is grafting experiments using wild-type plants and the NARC mutant. So here you can see the different combination, but the main message of this experiment is that NARC receptor is required to control the number of nodules under low phosphate condition. Here you can see that when the NARC receptor is mutated, particularly in the upper part of the plant, so the plant will keep forming about the same number of nodules independently of the phosphate status of the plant. So we did this experiment for Phaseolus vulgaris and also for soybean, and we observed basically the same behavior. And the interesting point is that if you remember, this picture, so the patchy pattern under optimal phosphate condition, the absence of this pattern under low phosphate condition in wild-type plants. But when we did the same experiment using the same transcriptional fusion, so you can see that this patchy pattern is also observed in plants growing under low phosphate condition, and I'm talking about the NARC mutant. So with this data, we are informing that at least the NARC receptor is really important to control the number of nodules under low phosphate condition. With all of this data, we have this model. This is under non-symbiotic condition, but under low phosphate condition. So basically, PHR is activating the expression of NIN. So we don't know what target genes NIN has under this particular condition. So the same PHR activate TML. We don't know so far the target proteins, but we are hypothesizing that in combination, NIN and TML are required for the plant adaptation to phosphate deficiency. And this is one of the questions that Ana Belén Mendoza, who is working in the lab with me, so is answering. And now I'm hoping that Mario and Jesus will organize another symposium, and maybe Ana Belén will present all of this data. But also I want to mention that there is reports that PHR is important in the nodule to sustain the demand of phosphate. So PHR can behave as a positive element by promoting the transport and translocation of phosphate to the nodule and sustain the high amount of energy. But also based on our data, so NARC or part of the ion is activating TML, blocking the activity of NIM, and as a result, inhibiting the nodule formation. So there are also, based on our transcriptomic data, that there is an additional negative regulator that will also affect the expression of key symbiotic genes required for nodule formation. So far with this data, we are trying to convince the community that phosphate levels are also important to sustain this symbiosis with rhizobia. So my last slide, I just want to take a few minutes to thank the people who make possible this data, and particularly members of my lab. And I want to thank Ana Belén, who came to my lab last year and took lead with this project. Now, hopefully, it will be accepted soon. But also my friend and collaborator, Damian Formey, who is also helping us with all the transcriptional analysis of our data. Obviously, Luis Cárdenas, that we have several discussions like, yes, phosphate is important, but we need to do more experiments for that. Ale Covarrubias, that also give us a key suggestion to answer these experiments. Very recently, Martina Reed from Germany joined efforts to answer this question, but in Lotus. So we have many genetic resources. That's the reason. But the beauty of Martina, she spent a lot of time as postdoctoral researcher trying to understand how PHR regulates the responses to phosphate deficiency, but also has a huge background on root nodule symbiosis. So I'm really optimistic that in a few years, we will have a bigger picture of the genetic components controlling this symbiosis under low phosphate condition. Jean-Michel Anet from University of Wisconsin, Pierre-Marc from the LRSB laboratories, and obviously who make this possible, our funders. So the GAPA, PAPIT, and the joint projects with the DFG. So now DFG is also financing our research on low phosphate conditions. The normal PAPIT for those working on in UNAM, and also the Secretaría de Ciencia, Humanidades, Tecnología e Innovación. And thanks all of you to spend some boring minutes with my talk and trying to convince you that phosphate is important for root nodule symbiosis. Thanks very much.
Thanks a lot, Osvaldo. Very
Thanks.
good. Let me, here with Danny.
Hi, thank you for the talk. I have a naive and conceptual question. So I don't know much about plants, but I understand that you're trying to convince us that phosphate is important, right? But how, this is why it's conceptual, but how do you define something that is important for symbiosis and something that is just because you mentioned that how expensive it is for the plant to go into symbiosis? So wouldn't it make sense that if the plant is stressed, under a lot of stress, it will have less nodules? So couldn't we basically say the same of, say, drought? Like this, I don't know if this is true, but any extreme stress for the plant, someone could make an argument that this is very necessary for symbiosis.
Yeah, I mean, I'm really lucky because if, for instance, if we talk about drought, so the responses to drought is totally different. So most of the negative effects comes in the mature nodules. So it depends where the stresses start. So, but the genetic components, there is work also in drought because now it's very important for climate changes. So it is completely different. So the genetic machinery is completely different. So it basically the plants decides to modify the root architecture to find water in the soil. But based on the data available, it's not involved in the signal transduction per se. But in the case of phosphate, it is involved in the signal transduction per se. So that's why I'm lucky.
Yeah, so just to make sure I understand, the argument is that because you can perfectly connect mechanistically, the phosphate sensing to the endosymbiotic pathway.
Exactly.
That's your argument. Okay.
Exactly.
Yes.
You want to go first? Okay. Thank you, Osvaldo. It was a very nice talk.
Thanks.
It's like every time you get a new piece of evidence, now I'm convinced. So
Thank you.
I'm going to make just two questions. So I was expecting, like, when you overexpress the phosphate response factors, you could make it able to modulate better independently of the phosphate level.
Yeah.
But it was completely
It wasn't the case.
opposite, you know?
Yeah.
And then you have the silencing, and then you have
Exactly.
the increase.
Exactly.
It's
Yeah,
like-
but now the point is, if we remove PHR, so the plant will say, okay, guys, so I'm able to perfectly interact with rhizobia, but I'm not able to cope with phosphate deficiency because PHR is a key component for plants to properly adapt to phosphate deficiency. So now, I mean, the idea is that we have to do is control the spatial temporal activity of PHR. I mean, we need to investigate more in details how and where PHR is expressed in basically in each step of the symbiosis to try to modulate according to the plant phosphate status.
Okay. And then my second question, it has to see with the point that you just made, that somehow the phosphate response could be also able to determine what kind of symbiont or what kind of microorganisms is going to be growing in the rhizospheres. A couple of years, there was a very nice paper describing that when you have low phosphate, you have this phosphate response activation, and then you increase the expression of one of the RALF peptide.
Yes.
And that RALF peptide is able to decrease the activity of the pheronial receptors,
Exactly.
and then less ROS is being produced in the rhizospheres. And now the new bacteria that are coming out in the microbiome are the ones that are able to provide that phosphate to the plants.
Exactly.
So that's a very clever experiment in which you can see that effectively phosphate response is able to modulate the microbiome in the rhizospheres.
Exactly. So there is a beautiful paper from Jeff Dungle group demonstrating that PHR is a key regulator of the plant pathogen interactions. But also we know that PHR is controlling the production of different secondary metabolites that are important to shape the root microbiome, for instance. So again, PHR, but also the phosphate status, to me, is a critical point for the plant to decide. So if I need phosphate, and most of my developmental process or communication with other microbes require energy. So first, I will satisfy my phosphate needs. Once they are satisfied, so I can start talking with the rest of the community. But if I don't have phosphate, so I'm sorry, I will not interact with you unless you provide me.
Right.
If you will demand me phosphate, so I mean, I'm trying to think as a plan. I mean,
so do we know if the phosphate response is expressed in the infected or uninfected cells? Do we have that level of
there?
In both?
Yes.
Okay, thanks.
Thank you for your presentation. I have two questions on EPP1. So I find this a very intriguing protein, and you mentioned that you now know it is phosphorylated by CMYK or DMA2.
Yes.
And if I remember, when you silence EPP1, it does not affect mycorrhizal fungal interaction. So how does it fit?
So in the first manuscript published in 2019, so we got a very weak phenotype, just because the different levels of EPP1 by RNA silencing. But after that, so I came back to Jean-Michel lab, and we generated unstable transgenic lines, and we did microrization assays, and basically it's black and white. So, and Pierre-Marc, so he's working with Marcantia, so he's a Marcantia lover. So basically, if EPP1 is not present, there is no interaction with mycorrhiza. So the explanation that I can provide you today is because the effect of RNAi roots. But now with the stable lines in Medicago and in Mercantia, so yes, it is required for microization.
Okay, good. That's nice. So you also mentioned that EPP1 is involved in the generation of some secondary messengers that will allow linking basically the membrane to the nucleus in terms of signaling. And when the initial work on mevalonate was published, I found this very intriguing, and it was shown that mevalonate was sufficient even in a HEK cell, so in an animal cell, upon expression of DMI1, to induce calcium spiking. So this will argue against the need of additional secondary messengers.
This is a very interesting point. What I can tell you today without telling names.
We can do it later.
So we did metabolomics in Medicago and also in Mercantia. And we found that when we modulate the expression of EPP1, the precursors of mevalonate are also affected. So that's why we came with the conclusion that EPP1, it is important for mevalonate, maybe for another metabolites derived from the precursors of mevalonate, but at least is indicating that EPP1 even is acting upstream of HMBR1 that Jean-Michel published in PNS some time ago. So he's sort of happy because he's involved with EPP1 too, but not that happy because it is very likely that maybe EPP1 is more important for that HMBR1. I mean, he's happy because he's collaborator, but that's why I say, okay, let's work together. Otherwise, we have another competitor and a strong competitor.
Good. I'm looking forward to see the paper. Thanks.
Thanks.
Congratulations.
Thanks. First, I have to say something. I read all your paper when I was a student, and I want to, at some point, say thanks for all the knowledge.
Drink something after the meeting. So I really agree with you that phosphate should be very important for the symbiosis. Just to remember, every cell needs DNA
Exactly.
And DNA needs phosphate.
Exactly.
So nodules are highly mitotic.
Sure, totally right.
And need phosphate. But I see a problem too. In your slide where you describe the mechanisms for modulation, I did not see any phosphate-related gene. Why? What do you think is going on that something is missing here?
Yes, totally right. So I have to admit, so this is the beginning. So I was trying to get as much evidence possible. So now with my collaborator in Germany, we want to fill up these gaps. So I mean, I have to also mention that I want to attract more funding from the former CONACYT because we want to know, like Luis Herrera did, like PHR is also controlling the chromatin remodeling. So we want to know if PHR is controlling, but also what other phosphate responsive genes are required for this symbiosis. So yes, I have to admit, this is just a small piece of the big puzzle. And I hope in 5 years, we have a bigger picture. I will not say the complete picture, but more detailed picture about the mechanism, how all the machinery related to phosphate deficiency, which part is important, which part is not important for the symbiosis.
Yes. Regarding this point, you focus on PHR1, but at least in Arabidopsis, there are two transcription factors.
Yes.
PHRL.
Yes, and the PHR-like proteins. This is a very interesting question. So now we are working with the PHR-likes protein, and we have now evidence with our collaborator that PHR-likes, at least the number 1, it is crucial to regulate the number of nodules. I mean, I'm not presenting this because I didn't generate this data, but I can share with you that the PHRLs, which both interact and make possible to activate the phosphate response, I mean, the responses to phosphate deficiency, yes. So at least another small piece of the puzzle is there. So the PHR-like, it is important.
Well, I think that the door is open and the coming weeks, years, I don't know, should be very fruitful to you. Thank you.
Thank you.
Another question?
Hi, Osvaldo. Thank you.
Hi, Sarita.
Can you imagine all of these scenario in the future, including more or less of this information with mycorrhiza?
Yes,
And the other one, mycorrhiza and rhizobium at the same time?
yes. So this is something that Ana Belén is working. So she will do some mycorrhization assays. For instance, with the plants with overexpression or silence in NIN to see if NIN is important for mycorrhization, but what happens if we put in an interaction? Some people will say, yes, if we inoculate plants with mycorrhizae along with rhizobia, we will solve this issue, but I would say it's not the case for all the legumes. Maybe for these 3 legumes will work because all the times are obtaining carbon to sustain any interaction, let's say. But in the case of Phaseolus or those important crop plants that have a very short life cycle, they cannot sustain many interactions at the same time. So that's why we want to know how the plant responds to phosphate deficiency. And the other question that we want to answer at some point is, can we develop a synthetic community able to help the plant to properly interact with rhizobia under low phosphate condition and make possible to harness all the ecological benefit of this symbiosis?
Thank you. And one thing more.
Sure.
Your model should be again being. You are going to continue with being as a model?
Yes. Yes. I mean, sorry, Jesus, but that's why Martina Reed will join me with Lotus. But I mean, as Luis said, I really love enfrijoladas. I really love frijoles with chicharrón. Yes, we need more beans.
You can eat the food and then make the experiments with genomics too.
Yes.
Okay. I think we are on time, but at the end now we will first. Thank you so much.
Thank you.
Les pedimos que se esperen un momento antes de ir al jardín a disfrutar pequeños canapés. Les den 10 minutos a la comisión que analizó los pósters para hacer la premiación y la clausura oficial aquí mismo en el auditorio y después ya vamos para allá. Entonces les pedimos un momentito, ¿vale? Muchas gracias.
Emocionados y ya con hambre además. Entonces, bueno, una de las cosas, digo, obviamente que es difícil siempre decidir, ¿no? Cuando hay tantos aspectos y la mayoría o todos los trabajos realmente muy, nos dejaron muy satisfechos en el sentido de que están muy completos, que la gente estuvo ahí, que los explicaron, etcétera. Entonces sí ha sido difícil y aquí está todo el comité, que ahorita vamos a decirles exactamente el nombre. Vamos a nombrar a 3, no quiere decir que no haya ninguna jerarquía, obviamente son 3 pósters que vimos muy completos. Y creo que el primero, bueno, para mencionar el primero, es el número 7. El póster número 7.
póster número 7, el título tiene Expanding A1, a cell wall modifying enzyme, is crucial for efficient intra and intercellular symbiotic infection in Lotus japonicus. First authors. There are two first authors, I guess. So it's Monroy Morales y Ivette García. A ver, ahora Claudia. No, que suban. Ok. 8.
Y ahora tenemos el segundo premiado. Es el número 8 y es Irán Tapia Vázquez. Y el nombre del póster es Genome Anagisha adiliensis, NG1, and its mutualistic interactions with different families. Congratulations. Bueno.
Representación de
Sí, ok, Carla. Muchísimas felicidades, Carla. Sí, también. Y bueno, representación de la...
And the third one is for
post number 28.
yeah. Okay, sorry about this. No. It's for the tension, you know, the drama of the yeah. Okay, but sorry, I remember the number. We don't know the title exactly, but it's poster number 28 for it's for the tension, you know, the drama of the yeah. And all the characterization of the phosphate soluble lysine strains. There were no particular order 1 to 3. Is all of them have their merit, even the ones that couldn't be in these 3 first places. But I mean, it was very, very, very difficult to select. And all the characterization of the phosphate soluble lysine strains. There were no particular order 1 to 3. Is all of them have their merit, even the ones that couldn't.
Les pido un aplauso especial a Mario y a Jesús. Por favor. Porque tuvieron la iniciativa y todo su esfuerzo. Y finalmente va a ser pues más breve que la inauguración. Un aplauso especial. Declaro oficialmente clausurado el evento. Y muchísimas gracias a todos por haberlo hecho posible. Hasta pronto, que tuvieron la iniciativa y todo su esfuerzo. Y finalmente va a ser pues más breve que la inauguración.